All right, so you know how we love to really get into the nitty-gritty of things here on the deep dive, right? Well, today we're diving headfirst into one of the most fundamental aspects of biology, the structure and function of microbial cells.
That's right. We're talking about those tiny but mighty organisms that rule the world, from the simplest bacteria to the more complex eukaryotes.
And we've got a fantastic chapter summary to guide us through this microscopic universe.
It covers a lot of ground, from the basic architecture of these cells to the mind-blowing mechanisms that allow them to thrive in every corner of the planet.
I mean, we're talking about organisms that can survive in boiling hot springs, in the freezing depths of the ocean, even in highly acidic environments. It's amazing.
It really is. And what's even more fascinating is how recent advances in technology, like cryogenic electron tomography or cryo-ET, are giving us a whole new level of understanding.
Cryo-ET. That sounds incredibly advanced. What is that exactly?
Imagine taking a cell, flash freezing it in its natural state, and then using a powerful electron microscope to create a 3D image of its internal structures at near atomic resolution. It's like taking a peek inside a living cell without disturbing it.
Wow, so we're talking about seeing the actual molecules and structures that make up these cells.
Exactly. And the things we're discovering are overturning some of our previous assumptions. For example, cryo-ET has revealed that a very common ocean bacterium, Candidatus pelagibacter ubique, has a surprisingly large periplasm.
The periplasm, that's the space between the inner and outer membranes in gram-negative bacteria, right?
Right. And we're also finding that the cytoplasm itself, which we used to think of as a kind of homogeneous soup, actually has distinct organized regions. So this technology is really changing our view of the microbial world.
So let's dive into the details. Our chapter summary starts with the cell envelope. It sounds like it's much more than just a simple outer layer.
It is. The cell envelope is the interface between the cell and its environment, and it plays a crucial role in a whole range of functions.
Like what?
Well, it controls what goes in and out of the cell, like nutrients and waste products. It helps the cell maintain its shape and provides structural integrity. It acts as a protective barrier against environmental stresses and predators.
And it's even involved in interactions with other cells, right? Like forming biofilm.
Exactly. So it's a dynamic, multifunctional structure that's essential for microbial survival.
Okay, so what makes up this cell envelope? The chapter summary mentions the cytoplasmic membrane as the innermost layer.
Right. The cytoplasmic membrane, or plasma membrane, is found in all living cells, both prokaryotes and eukaryotes.
It's that classic phospholipid bilayer structure, right? The one with the hydrophilic heads facing outward and the hydrophobic tails tucked in the middle.
Precisely. That arrangement is key to its function as a selectively permeable barrier. It allows the cell to control what enters and exits the cytoplasm.
And embedded within this bilayer are various proteins that carry out important functions.
Right. There are integral proteins that span the entire membrane, acting as channels or transporters, and there are peripheral proteins that are more loosely associated with the membrane surface, often interacting with the integral proteins.
Now, the chapter summary points out that while both bacteria and archaea have cytoplasmic membranes, there are some significant differences in their composition.
Yes, the archaeal membranes are really interesting. They use different types of lipids than bacteria and eukaryotes.
Like what?
Well, for one, their lipids have isoprenoid chains instead of fatty acids. And these chains are linked to glycerol by ether bonds instead of ester bonds.
And why is that significant?
It makes their membranes much more resistant to extreme conditions, like high temperatures and acidity, which is why many archaea can thrive in those environments.
So it's like they've evolved a more robust membrane structure to cope with those harsh conditions.
Exactly. They've also got some unique lipids, like diphosphoglycerol tetraethers, which can form a lipid monolayer instead of a bilayer.
A monolayer, so a single layer of lipids spanning the entire membrane.
That's right. And some archaea even incorporate ring structures into their lipids, like Archaea.
So same basic concept as the bacterial membrane, but with some significant modifications for those extreme lifestyles.
Absolutely. It highlights the diversity and adaptability of microbial life.
Okay, beyond acting as a barrier, what other important functions does the cytoplasmic membrane perform?
Well, it's a key player in energy conservation. That's where the proton motive force is generated.
The proton motive force, that's that electrochemical gradient that's used to power so many cellular processes, right?
Exactly. And the membrane is also crucial for anchoring various proteins involved in transport, bioenergetics, and even sensory systems.
Right, because most substances can't just diffuse across the membrane on their own. They need help from those specialized transport proteins.
Exactly. And those proteins are incredibly selective and efficient, often transporting molecules against their concentration gradients.
Meaning from an area of low concentration to an area of high concentration, which requires energy.
Precisely. And the chapter summary breaks down these transport systems into three main classes.
Let's go through them. First up, simple transport. What's the defining feature of this system?
Simple transport uses a single membrane-spanning protein to facilitate the movement of a solute across the membrane.
And the energy for this process comes from the proton motive force.
Right. We see examples like symporters, which move a solute along with a proton in the same direction, and antiporters, which exchange a solute for a proton in the opposite direction.
So it's a direct coupling of solute transport to the energy stored in that proton gradient.
Precisely.
Okay, next we have group translocation, which sounds a bit more complex.
It is. In group translocation, the substance being transported is chemically modified during the process, which requires energy from a high-energy organic compound, like phosphonol pyruvate or PEP.
And the classic example is the phosphotransferase system in E. coli, which is responsible for transporting sugars.
Right. As the sugar is transported across the membrane, it gets phosphorylated, which prevents it from leaking back out, and also helps maintain a concentration gradient that favors further uptake.
So it's like a one-way ticket with an added processing step.
Exactly.
And finally, we have the ABC transporter systems, which sound even more sophisticated.
They are generally more complex, typically involving three components: a substrate binding protein, a transmembrane channel, and an ATP hydrolyzing protein.
And these systems use ATP directly as their energy source.
That's right. The substrate binding protein has a high affinity for the specific nutrient it transports, even at very low concentrations.
So they're like super efficient scavengers.
Exactly. They're especially important in nutrient-poor environments.
Okay, so we've got these three different transport systems, each with its own mechanism and energy source, working to get essential molecules into the cell.
That's right. It highlights the diversity and ingenuity of microbial adaptations for acquiring nutrients.
Now, let's move on to the next layer of the cell envelope, the bacterial cell wall. And the spotlight here is definitely on peptidoglycan.
Absolutely. Peptidoglycan is a unique and essential component of the bacterial cell wall.
It's responsible for providing structural integrity and protecting the cell from osmotic lysis.
Exactly. You see, bacteria typically have a higher solute concentration inside their cells compared to their surroundings.
So water tends to flow into
Okay.
the cell by osmosis.
Right. And without a strong cell wall, the cell would swell and eventually burst.
So peptidoglycan is like a molecular chainmail that prevents the cell from exploding.
That's a great analogy, and it's this unique structure that makes it such an effective target for antibiotics.
Tell me more about the structure of peptidoglycan.
It's a polymer made up of long chains of two alternating sugar derivatives, N-acetylglucosamine and N-acetylmuramic acid.
And these chains are cross-linked by short peptide chains.
Right. And the specific way these peptide chains are cross-linked differs between gram-positive and gram-negative bacteria.
Ah, yes, the famous Gram stain. It all comes down to the thickness of that peptidoglycan layer.
Exactly. Gram-positive bacteria have a much thicker peptidoglycan layer, up to 90% of their cell wall, which allows them to retain the crystal violet stain.
While gram-negative bacteria have a thinner peptidoglycan layer sandwiched between two membranes.
Right. And they don't retain the crystal violet stain.
So this simple staining technique can tell us a lot about the structure of a bacterium's cell wall.
It certainly can, and it's a fundamental tool in bacterial identification and classification.
Now, the chapter summary mentions teichoic acids and lipoteichoic acids, which are found embedded in the peptidoglycan layer of gram-positive bacteria. What are their roles?
They're acidic polymers that contribute to the overall rigidity and negative charge of the cell wall. Lipoteichoic acids, in particular, span the peptidoglycan layer and are anchored to the cytoplasmic membrane.
So they're like additional structural reinforcements and also play a role in anchoring the cell wall to the membrane.
That's right.
The chapter summary also mentions lysozyme and penicillin, two substances that can disrupt peptidoglycan. Why are they significant?
They highlight the critical importance of peptidoglycan for bacterial survival. Lysozyme is an enzyme that breaks down the glycosidic bonds in peptidoglycan, essentially weakening its structure.
And penicillin is an antibiotic that inhibits the formation of those peptide crosslinks.
Exactly. Both of these actions compromise the integrity of the cell wall, making the bacteria susceptible to osmotic lysis.
So that's why they're effective antibacterial agents.
Precisely.
Okay, let's talk about the outer membrane in gram-negative bacteria. What makes it unique?
It's an additional lipid bilayer located outside the thin peptidoglycan.
And it's separated from the peptidoglycan by the periplasm.
Right. And this outer membrane contains lipopolysaccharide, or LPS, which is a major virulence factor in gram-negative bacteria.
LPS, that's the one that can trigger a strong immune response in animals, right? It can even lead to septic shock.
Exactly. It's a powerful molecule, and it's composed of three main parts. Lipid A, which anchors it to the membrane and acts as the endotoxin, the core polysaccharide, which is relatively conserved, and the O-specific polysaccharide, which varies greatly between strains.
So this O-specific polysaccharide is what our immune system uses to recognize different strains of bacteria.
That's right. It's like a fingerprint for the bacteria.
Now, the outer membrane is described as being more permeable than the cytoplasmic membrane. How is that possible with another lipid bilayer?
It's due to the presence of porins, which are transmembrane protein channels that allow the passive diffusion of small hydrophilic molecules.
So they act like molecular sieves, letting certain molecules through while keeping others out.
Exactly. Some porins are nonspecific, while others are more selective, transporting specific nutrients.
Well, what about the periplasm? It's not just an empty space, is it?
Not at all. It's a busy compartment filled with a variety of proteins involved in nutrient degradation, transport, chemotaxis, and cell wall synthesis.
So it's like a staging area for molecules entering and exiting the cell.
That's a good way to think about it.
The chapter summary also mentions bron lipoprotein. What's its role?
It acts as a molecular anchor, connecting the outer membrane to the peptidoglycan layer, providing stability to the entire cell envelope.
Okay, so we've covered the gram-positive and gram-negative cell envelopes in detail, but the chapter summary emphasizes that there's even more diversity in microbial cell envelope structures.
Oh, absolutely. Those two models are just the tip of the iceberg.
Yeah, but what about those S layers? They sound pretty interesting.
S layers are crystalline layers made of protein or glycoprotein that are found in many bacteria and almost all archaea.
And they form the outermost layer of the cell envelope.
In many cases, yes. They can act as a protective barrier, molecular sieve, and they can even be involved in cell surface interactions.
So they're like an additional layer of armor for the cell.
In a way, yes. And in some archaea, they actually serve as the primary cell wall, providing structural support.
And then there's pseudomurane, found in some archaea. How does it compare to peptidoglycan?
It's structurally similar, but it has some key differences that make it resistant to lysozyme and penicillin.
So those antibiotics wouldn't work against archaea with pseudomurine cell walls?
That's right.
And what about microbes that lack a cell wall altogether?
There are some bacteria, like mycoplasmas, and some archaea, like thermoplasma, that don't have a rigid cell wall.
So how do they survive without that structural support?
They have unusually tough cytoplasmic membranes, sometimes with sterols that provide additional rigidity.
So they've adapted to survive without that external layer of protection.
Exactly.
Finally, the chapter summary mentions some archaea, like Ignococcus, that have a unique outer membrane different from the one found in gram-negative bacteria.
Yes, Ignococcus is a fascinating example. Its outer membrane is primarily composed of archaeal lipids and lacks lipopolysaccharide.
So another variation on the theme of outer membrane structure.
Exactly. It highlights the incredible diversity and evolutionary ingenuity of microbial life.
Okay, let's move on from the cell envelope and explore some of the other structures found on the surface of microbial cells.
Sure. There are capsules and slime layers, pili, and even grappling hook-like structures called hami in some archaea.
Capsules and slime layers, those are those gooey coatings that some bacteria have, right?
That's right. They're made of polysaccharides or proteins and can play various roles.
Like what?
They can help bacteria attach to surfaces, which is important for biofilm formation. They can protect the bacteria from desiccation and from being engulfed by immune cells.
So they're like a shield for the bacteria.
In a way, yes. And they can also help trap nutrients in water.
Now, what about pili? They sound a bit more structural.
Pili are thin, filamentous appendages made of protein. There are different types of pili, each with its own function.
Like what?
Well, fimbriae are shorter pili that are primarily involved in adhesion, helping bacteria stick to surfaces or other cells.
And then there are the longer pili, like the sex pili, which are involved in conjugation.
Right. Conjugation is the process of transferring genetic material between bacteria.
So it's like bacterial mating.
In a way, yes. And there are also type IV pili, which are involved in twitching motility, a type of jerky movement across surfaces.
So these little pillies are quite versatile.
They are. And some bacteria even have electrically conductive pili called nanowires, which are thought to play a role in energy metabolism.
Wow, that's incredible. And what about those hammy you mentioned?
Hammies are found in certain archaea that live in anoxic sediments.
And they're like grappling hooks.
That's right. They have a barbed, hook-like structure at the end, which allows them to attach to surfaces and to each other.
So they're like anchors for the archaea.
Exactly. And they can form large interconnected networks, which might be important for nutrient scavenging in those nutrient-limited environments.
Okay, now let's move inside the microbial cell and talk about inclusions. These are those internal storage compartments and specialized structures.
Right. They're like miniature organelles that carry out specific functions.
And the JAPA summary mentions they're typically enclosed by a single-layer protein membrane.
That's right. It separates them from the rest of the cytoplasm.
So what kind of inclusions are we talking about?
Well, there are carbon storage polymers like PHB and glycogen, which store energy and carbon for later use.
Like a pantry for the cell.
Exactly. And there are polyphosphate granules, which store inorganic phosphate for use in nucleic acid and phospholipid synthesis.
And then there are sulfur globules found in sulfur-oxidizing bacteria.
Right. They store elemental sulfur, which is an intermediate product of their metabolism.
And some bacteria even have carbonate minerals inside their cells.
It's a process called biomineralization. The exact function of these minerals isn't fully understood, but they might be involved in buoyancy regulation or carbon sequestration.
And then there are gas vesicles, which sound pretty cool.
They're gas-filled structures that provide buoyancy to aquatic microorganisms, allowing them to move up and down in the water column.
So they're like tiny flotation devices.
Exactly. And finally, there are magnetosomes, which are truly remarkable.
Those are the ones that contain magnetic iron oxide crystals, right?
That's right. They allow bacteria to orient themselves along the Earth's magnetic field lines.
So they're like internal compasses for the bacteria.
Exactly. And it's thought to help them navigate towards optimal oxygen concentrations.
Okay, that's a pretty impressive array of inclusions. These microbes are really well equipped for survival. Now, let's talk about endospores. These are the ultimate survival specialists, right?
Absolutely. Endospores are dormant, highly resistant structures formed by certain gram-positive bacteria.
And they're incredibly resilient, able to withstand extreme temperatures, radiation, and even harsh chemicals.
That's right. They can survive for incredibly long periods, even millions of years.
So how do they achieve this remarkable level of resistance?
It's a combination of factors. The core of the endospore is dehydrated, which helps stabilize its DNA and proteins.
So it's like they've dried themselves out to survive.
In a way, yes, and they contain a high concentration of dipiclinic acid, which also helps protect their DNA.
And their DNA is further protected by small acid-soluble proteins, or SASPs.
Right, and then they're encased in multiple protective layers. The spore coat, the cortex, and sometimes an exosporium.
So it's like they've built themselves a fortress.
Exactly, and when conditions become favorable again, they can germinate back into actively growing cells.
So it's like they've hit the pause button on life until things get better.
That's a good analogy.
Okay, now let's talk about how these microscopic organisms move around. The chapter summary focuses on swimming and gliding motility.
Right. Motility is crucial for many microbes, allowing them to seek out favorable conditions and avoid harmful ones.
So let's start with swimming motility, which is usually powered by a flagella in bacteria and archaea in Archaea.
Those are those whip-like appendages that propel the cell through liquid environments.
And they rotate, right?
Exactly. The rotation is powered by the proton motive force in bacteria and by ATP hydrolysis in some archaea.
And the direction of rotation determines the direction of movement.
Right. Counterclockwise rotation usually results in a straight run, while clockwise rotation causes the cell to tumble, changing direction.
And there are different arrangements of flagella on the cell surface, right?
Yes, there's polar flagellation, where the flagella are located at one or both ends of the cell, and peritricious flagellation, where the flagella are distributed all over the cell surface.
So different arrangements for different modes of swimming.
Exactly. And the flagella themselves are pretty amazing structures.
How so?
They're composed of a filament, a hook, and a basal body, which is the motor that drives the rotation.
And the filament is made up of flagellin protein subunits, right?
That's right. And the hook acts as a flexible coupling between the filament and the basal body.
And the basal body is embedded in the cell envelope.
Exactly. It's a complex structure with a rotor and a stator, and it's incredibly efficient at converting energy into rotational motion.
Okay, so that's swimming motility. Now what about gliding motility?
Gliding motility is a smooth, continuous movement across surfaces without the use of flagella or archaea.
And it's not fully understood how it works.
Not entirely. There are several models, but the exact mechanisms are still being investigated.
But it's observed in a variety of bacteria and archaea.
That's right. Some glide on a layer of slime, others use surface adhesion proteins, and some might even have internal motors that interact with the surface.
So a variety of solutions for achieving this type of movement.
Exactly. And it's another example of the diversity and adaptability of microbial life.
Now, microbial movement isn't always random. They often move in response to specific stimuli, like chemicals, light, or oxygen. This is called taxis.
That's right. Chemotaxis is the movement towards or away from chemical gradients.
So how do they sense these gradients and adjust their movement accordingly?
They have chemoreceptors, which are membrane proteins that bind to specific chemicals.
And these receptors send signals to the flagellar motor, telling it to rotate in a particular direction.
Exactly. So if a bacterium senses a higher concentration of an attractant, it will tend to run for longer periods in that direction.
And if it senses a repellent, it will tumble more frequently to change direction.
Right. It's called a biased random walk.
So it's not a direct targeted movement, but rather a series of adjustments based on the chemical cues they're receiving.
Exactly. And it allows them to effectively navigate towards favorable conditions.
And there are other forms of taxis besides chemotaxis, right?
Yes. There's phototaxis, movement in response to light, aerotaxis, movement in response to oxygen, and even magnetotaxis, using those magnetosomes we talked about earlier to orient themselves along magnetic field lines.
So these microbes are really good at sensing their environment and responding accordingly.
They are. It's essential for their survival.
Okay, we've covered a lot of ground on prokaryotic cells. Now let's shift our focus to eukaryotic microbial cells, like fungi and protists. What are the key differences?
Eukaryotic cells are much more complex than prokaryotic cells. They have a membrane-bound nucleus that contains their DNA, and they have other specialized organelles.
So they're more compartmentalized.
Exactly. And this allows for a greater level of complexity in their structure and function.
Let's start with the nucleus. What's its main function?
The nucleus is the control center of the cell. It houses the DNA, which carries the genetic instructions for the cell.
And it's enclosed by a double membrane called the nuclear envelope.
Right. And this envelope has pores that regulate the movement of molecules between the nucleus and the cytoplasm.
So it's not completely isolated from the rest of the cell.
No, there's constant communication between the nucleus and the cytoplasm.
And inside the nucleus, the DNA is organized into chromosomes.
Exactly. And those chromosomes are made up of DNA tightly wound around histone proteins.
So it's like a very organized filing system for the genetic information.
That's a good way to think about it.
And there's also a region within the nucleus called the nucleolus.
Right. The nucleolus is where ribosomal RNA is synthesized and ribosomes are assembled.
So it's like the ribosome factory of the cell.
Exactly.
And when eukaryotic cells divide, they undergo mitosis. How does that differ from binary fission in bacteria?
Mitosis is a more complex process that involves replicating the chromosomes and then separating them into two identical daughter nuclei.
So it ensures that each daughter cell receives a complete set of chromosomes.
Exactly. And it's divided into several phases: prophase, metaphase, anaphase, and telophase.
And then there's cytokinesis, the division of the cytoplasm.
Right. That results in the formation of two separate daughter cells.
Now let's talk about mitochondria and chloroplasts, the energy-generating organelles of eukaryotic cells.
Mitochondria are responsible for aerobic respiration, the process that converts nutrients into ATP, the cell's energy currency.
And chloroplasts are found in plants and algae and are responsible for photosynthesis.
Exactly. And both of these organelles are thought to have originated from bacteria that were engulfed by ancestral eukaryotic cells.
That's the endosymbiotic theory, right?
That's right. And there's a lot of evidence to support it.
Like what?
Well, mitochondria and chloroplasts have their own DNA, which is circular like bacterial DNA.
And they also have their own ribosomes, which are more similar to bacterial ribosomes than eukaryotic ribosomes.
Exactly. And they have a double membrane, which is consistent with the idea of being engulfed by another cell.
So these organelles are like little remnants of ancient bacteria living inside eukaryotic cells.
In a way, yes. And they're essential for the energy production and metabolism of eukaryotic cells.
Now, what about some of the other eukaryotic organelles, the ones that didn't arise through endosymbiosis?
Well, there's the endoplasmic reticulum, or ER, which is a network of membranes involved in protein synthesis, lipid metabolism, and detoxification.
And there's the Golgi complex, which modifies, sorts, and packages proteins for secretion or delivery to other organelles.
Exactly. It's like the cell's post office.
And then there are lysosomes, which break down waste materials and recycle cellular components.
Right. They contain a variety of digestive enzymes.
And finally, there's the cytoskeleton, which provides structural support and facilitates movement within the cell.
The cytoskeleton is made up of a network of protein filaments, including microtubules, microfilaments, and intermediate filaments.
And each of these filaments has a specific role in the cell.
That's right. Microtubules are involved in cell shape, motility, and chromosome movement. Microfilaments are involved in cell shape changes and muscle contraction, and intermediate filaments provide mechanical strength.
So the cytoskeleton is a dynamic and essential part of the eukaryotic cell.
It is?
And finally, eukaryotic cells can also have flagella and cilia, which are involved in motility.
But they're structurally different from the flagella found in bacteria.
How so?
They have a core of microtubules arranged in a 9+2 pattern, and they move by a bending or whip-like motion rather than rotation.
So another example of convergent evolution, where different structures have evolved to serve similar functions. Well, that was a whirlwind tour of the microbial world. We covered everything from the basic architecture of prokaryotic and eukaryotic cells to the amazing diversity of structures and adaptations they've evolved.
We discussed cell envelopes, surface structures, internal inclusions, endospores, motility mechanisms, and even the endosymbiotic theory.
It's incredible to think about the complexity and ingenuity that exists at this microscopic level.
It really is, and it highlights the importance of studying these tiny organisms, not just for their own sake, but also for their impact on our world.
From human health to the environment to biotechnology, microbes play a crucial role in countless processes.
They're essential for life as we know it.
And with that, we conclude our deep dive into the fascinating world of microbial cell structure and function.
Hopefully, this has given you a new appreciation for these tiny but mighty organisms.
And perhaps inspired you to explore this field further on your own.
There's always more to discover in the microbial world.
Thanks for joining us on this deep dive.
It was my pleasure.